The last class we talked about DNA replication, right? And the fact that is semi-conservative and the enzymes involved in DNA replication, how it makes copies of itself, which was very simplified and basic, but that's how it is. Continuing with that topic, today we're going to talk about the genetic code. Again, right, it's a simple topic, so we might be able to cover more ground. But let's get started with this first. The genetic code. And as the name suggests, what we're going to talk about is how the DNA functions as the molecule holding the information that is important for life. Right, so DNA holds the instructions for life, and it is made up of, right, nucleotide bases, which in this case are adenine, thymine, guanine, and cytosine, represented by those alphabets. And how this happens is, right, DNA codes for proteins, so the genes, right, genes are coded into proteins. One gene codes for one protein pretty much. And proteins are made of amino acids, right? So, and a single polypeptide, a protein is, just to highlight, I think I've mentioned this earlier as well, right? So a polypeptide is a polymer of amino acids, right? And a protein is a functional biological molecule made of amino acids. How are these two different? These two are different because it is possible that you may have a polypeptide chain that is not a protein, and that is because sometimes proteins are made up of multiple polypeptide chains. So the example is hemoglobin, right? Hemoglobin is the molecule that is responsible for carrying oxygen in your red blood cells, and it is actually made up of 4 polypeptide chains, right? So 4 polypeptide chains, 2 alpha globin, 2 beta globin chains come together to form one molecule of the protein that is known as hemoglobin, right? So a protein could be a single polypeptide chain, like the insulin protein, right? That hormone is a single polypeptide chain. It could be multiple polypeptide chains coming together to form a protein, right? So there is that difference between polypeptide and protein. Now, right, any case, proteins are made up of polypeptide chains, and those are made up of amino acids. And so what we have are 20 naturally occurring amino acids. And we have 4 nucleotide bases. So doing simple math, right, if one nucleotide base coded for an amino acid, you'd only have 4. So that's obviously not working out. If 2 nucleotide bases, right, or you say 4 squared, coded for an amino acid, you'd only get 16. So you're still missing out on 4 of them. So you go to the next step, right? you take 3 amino acids coding for a single, sorry, 3 nucleotide bases coding for a single amino acids. And right, this is known as the triplet code. So this is what was discovered to actually, take place. It's a triplet code, right? So there's 64 possibilities for these, these 4 alphabets to arrange. You only need 3 of them. And so, right, you've got more possible combinations than you've got amino acids. And so what that brings us to is that the triplet code in DNA, the triplet code is also known as a codon, right, in DNA, is redundant. And I don't like using that word, but that's the word they use. So it's called redundant. And the reason for that is when it was studied, you obviously have 20 codes for 20 amino acids, right? But then you've got some, other codes that code for the same amino acids, right? So for example, if I take a look at that, obviously I don't have it memorized. And so if you take a look at it and see what are the different possibilities that code for an amino acid called leucine. And so there are 6 possibilities, right? So if you take a look at the nucleotide bases, you have AAT and AAC and GAA and GAG, GAT and GAC, right? And so there are 6 codes for the amino acid leucine. Given by the short form of the first 3, leucine. Now, right, how does this help? We're calling it redundant, which is not, it doesn't sound nice, but it's actually it's a very protective feature. And the reason for that is, are you following along? Like, did you see how did you follow how I shifted from talking about the nucleotide bases to coding for amino acids? Does that make sense?
Yes, sir.
Okay. And so why is this redundancy helpful? It's helpful because, right, when DNA is being replicated in cells, right, point mutations occur often. Point mutations, right, are mutations. So they are changes in a single nucleotide base, right, at any point along the DNA. And so, right, if on there were only 20 codes for 20 amino acids, what would happen if you would had, for example, right, AAT, and right, it was changed to AAC, and so theoretically you would have a different amino acid, and that would be problematic. Why? Because the sequence of amino acids required for polypeptide chains, for functional usable polypeptide chains, is specific. It's not like the way you're arranging your clothes in your wardrobe and you can put any put them in any order and they're all the same, they're all, hanging on, hanging, and it doesn't matter, right? So it's specific, right? If you have, for example, a sequence of amino acids, you're starting with methionine, and then you've got, right, this, and then you've got proline. So let's just write pro for that. And then you've got leucine, and then you've got valine. And so, right, if you have something different, for example, you have, right, just this particular change, moving one amino acid, just switching these two, and that's going to completely change the polypeptide chain, the way the polypeptide chain folds. And so we've talked about the coordinate, the different foldings of the polypeptide chains, the primary, secondary, all that. And so this makes a huge difference, right? It's not the same thing, right? It might produce a faulty amino acid, might produce a non-functional amino acid, or it might even make something toxic and detrimental to the organism, the whole, in which this problem has occurred in that cell. But as you can see over here, right, in case of leucine, it makes no difference, right? So if the AAT changed to AAC, and then you've got GAA, GAG, GAT, GAC, it's all the same, right? Now this is an example where leucine has 6 different codes, right? Not all amino acids have that many. Some have 2, 3, and there are like 2 of them that only have a single code going for them. If I can just locate which ones those are, right? So those are methionine and tryptophan, right? So there are 2 amino acids which only have a single code that go for them, and those are methionine is TAC, and tryptophan is ACC. So these are the only 2 amino acids that have a single code. So if there's any change in these, obviously that would be problematic, but the rest have at least 2, let's say. And so if point mutations were to occur, they would not be as bad. Of course, we're taking a look at the third amino acid, like the third nucleotide base of the triplet code. The point mutation could occur anywhere, and still, right, it does give a degree of protection from mutations. So even if the mutation occurs, it's still coding for the same amino acid, and so you're still ending with the same polypeptide chain, which will lead to the same protein, and so functionality is not affected, and so the organism's health is not affected, right? That being said, there are mechanisms in place in the cells. If a mutation does occur, uh, there are attempts to try and correct it. So it's not like, oh, it happened, let's carry on, it's fine. No, it doesn't work that way. So there are attempts to uh, repair it, but if it's not repaired, this is like a safety net. Okay, what else? We've talked about the degenerate code. Well, it's right. Formally, this is like the redundancy is right. It's called the degenerate code. This is the formal name for all of this. Then, right, in all of these 64, there's another thing that was noticed. It was experimented, and conclusion reached that the DNA, like the amino acid sequence, the nucleotide sequence is non-overlapping. What do they mean by this? So I'll use the same example they've given. And what they did is that they studied this using mRNA, right? mRNA is a type of RNA that's taken from DNA when protein synthesis occurs, right? You're obviously familiar with that from perhaps your IGCSE. We'll come to it in a bit. And so what they take is the mRNA sequence, and for example, right, the mRNA sequence is UUUAGC. Now remember, mRNAs don't have thiamine, right? Instead of thiamine, they've got uracil. It's given with a U. Now, right, the UUU codes for phenylalanine. Now we talked about the triplet code, right? What does it mean by its non-overlapping? So if it were overlapping, right, you start off with the first three, and then what you do, you leave the first and you go with the next three, and then you go with the next three, and then, right, so there's overlap. You have the same two U's that were in the code for phenylalanine, but now you're looking at UUA, and then you take the last U and you have UAG, and then finally you've got AGC. So that is the overlap they're talking about, right? That the nucleotide bases, they are forming triplet codes, but they're not individual segments, right? So you're just taking U, and AGC. Now this is a theory, right? And so what they would individually code for, this would be in the mRNA, this would be leucine, and UAG is something called a stop codon, and then AGC is serine. Now, right, there are two problems with this. The first problem is that if it were overlapping, if it were the way that we are theorizing, then it although the benefit is that it becomes far more compact. Instead of having 12 nucleotide bases for these four, like three amino acids and a stop codon, you fit all of that into just six, right? So that's half, right? And so it's far more compact, and you expand it further, and so you see how much, like, compact it becomes compared to you spreading out the whole thing. So that is a benefit. So if I write that, the benefit is it's more efficient and compact, so to speak, right? You can carry more information in less space. But that's just one. What are the drawbacks? And they're more severe, right? One is it's limited. Why is it limited? For example, right, in a sequence like this, you, right, UUA is leucine and UUU is phenylalanine. And so, right, you are limited as to which amino acid can, sequence you can follow. So in this case, only leucine will be able to come out of phenylalanine. If I want serine to come in, it's not going to come in because I'm stuck with that double U to begin with, right? I can't have AGC coming in over here because it's overlapping, right? And so it's limited variation in the amino acid sequence. And obviously that's going to limit the, the kind of proteins, the kind of polypeptides that can form. So that's one drawback. And the second drawback, as you see in this, right, code, I'm getting like a stop signal in between. So UAC is coming in between, and so I can't control that, right? So for example, in this example, I'm getting unwanted codes in the middle. So unwanted or undesirable codes. Now, the experiment that was conducted, to check whether it's completely overlapping or partially overlapping or non-overlapping. And so what they did is that they actually made point mutations. And so if it were completely overlapping, for example, they mutated this U, right? And what they did was they turned it into a U, let's say G, and then, right, an AGC. And so this third U is present in in three of these. So if it were completely overlapping, right? So the experiment used E. coli bacteria, right, and made point mutations. And if it were completely overlapping, right, a single mutation would affect 3 amino acids. Because it's affecting 3 codes. If partially overlapping, right, it would affect only 2. How would it be partially overlapping? So uh, if uh, it did not go stepwise, it jumped one. So you went from UUU to UAC directly. You skipped one in between, right? And so that. But if it were non-overlapping, which is what was found, right, it would affect only a single amino acid. And so in their experiments with the point mutations, that's what they found, that any point mutation they made only affected a single amino acid in the entire sequence. And so, right, that actually makes it safer. It makes it bigger, but it makes it safer. And it also gives you a lot of freedom in as to which amino acid can be, like, again, you're not doing it yourself. It's happening naturally, but it removes that, limitation that would have been there if it were overlapping, whether partially or completely. Is that clear? Does that make sense?
Yes, sir. It does.
Okay. Have I missed anything? Uh, okay, so all right, some other stuff that they mention which will lead us into protein synthesis. So as we know that the DNA has functional sections called genes, and so the way it works is the DNA never leaves the nucleus. It's always in the nucleus. It's never been found outside the nucleus, except for the process of cell division, in which case the nuclear envelope breaks down. So you can't really say it's outside the nucleus in that case. That's a very specific case. And right, one of the strands is called the template strand. So it's a double helix, and so the template strand is transcribed into something called mRNA. Right, so it's an RNA molecule, and the M stands for messenger. right? So messenger RNA. So what's the difference between that, right? One of the differences is, right, it has a ribose sugar instead of a deoxyribose. So we talked about that earlier. And secondly is that the nucleotide bases are A, U, G, and C. It doesn't have a thymine, it has a uracil instead. We just mentioned that. And so the template strand is transcribed into the mRNA, and then the mRNA, right, goes to the cytoplasm, and there it attaches to a ribosome, right, which is an organelle and responsible for protein synthesis. And so protein synthesis occurs on the ribosome, right, using the mRNA. It's interesting to know that, right, 98% of the DNA is non-coding. So it's like, so it's not genes, right? And so these, right, they're non-coding, but then they've got other functions, right? So they might have sections of codes that regulate gene expression. So they turn genes on and off, right? And then they've got you know sections such as promoters, which are again, right, so I'll put that as an example. So such as a promoter region, so that's a particular sequence on the DNA that helps it in DNA expression, or there might be a repressor or might be something else of the sort, right? Binding sites for different proteins that are involved in gene expression. And so that's 98, right? So only 2%, right, of DNA has codes for proteins, right? So they've got codons for amino acids, right? Uh, and with that, we lead into chapter 2b6, which is DNA and protein synthesis. Any questions for what we've talked about so far?
No, sir.
Okay. So as you talked about RNA, there are different types of RNA, and right, they, like one of them at least is made from DNA, which we just talked about. So that's the messenger RNA, and that, so messenger RNA. Excuse me. And so its function is that it carries instructions for a polypeptide chain from the DNA in the nucleus to the ribosome in the cytoplasm. Now, talking about that further, right, it's written as mRNA for short, the M being lowercase. And right, so what happens is when a certain segment of DNA, a certain gene needs to be, coded for the, to make the protein, part of the DNA unwinds or unzips, right? And so you've got the two strands, there's a double helix, right? So there are two strands. There's a sense strand has the code, right? And then the complementary strand of DNA is called the antisense strand. And what happens is that this becomes the template for mRNA. Now, why does that happen? So, right, this needs a bit of illustration. And so, for example, you have the sense strand, right? So this is the sense strand. And the sequence on the sense strand goes like this. T, G, 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 C, G, G. So T, G, 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 C, G, G, A, T, 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 C, T, G, G. Right? And this is the actual code for the polypeptide. What are you going to have on the antisense strand? Right? And this guy becomes the template. So we'll see. So what you're going to have is the corresponding. So T gives you A, and then C, 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 G, C, C, and then T, A, 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 G, A, C, C. Have I made a mistake anywhere? A, C, 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 G, C, C, T, A, 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 G, A, C, C. Okay, so that's what you're getting. Now, why does this strand become the template for the mRNA, right? So what happens is, right, RNA polymerase binds, I'll write that down later, and it starts making the mRNA molecule. And so what's the code you're going to, that's going to show up over here, right? What's complementary to this? It's T. Now, right, we don't have thymine in mRNA, so you're going to get a U. G, 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 C, G, G, right? A, U, 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 C, U, yeah, G, G. Now, if you just take a look at it, this code is exactly the same as this code, except for the difference of the thiamine and the uracil, right? It's exactly the same. You've got all the guanines and the adenines and the cytosines in the same positions, right? And so what you end up with is an mRNA molecule that has the same nucleotide sequence as the sense strand, right? Which is the actual code for the polypeptide. And that's why the antisense strand is used as the template. Does that make sense?
Yes.
Okay. So now we have our mRNA molecule. Another thing to mention over here is something called the start and stop codons. Right, so as you mentioned, codon is like a triplet code of nucleotide bases that generally code for amino acids, but then there are a few that have a separate function, right? So there's something called a start codon, and what this does is it signals the point of transcription. So when the DNA is being transcribed into mRNA, right? Transcribe to mRNA, so that's called transcription. and that code on the sense strand is TAC, and it also codes for the amino acid methionine, which we mentioned earlier, right? So this is the only code. So every polypeptide starts with methionine. Now, once a protein is, it's completely made on the ribosome, the polypeptide chain is complete, it goes into the rough endoplasmic reticulum and then to the Golgi apparatus later on, and it undergoes post-processing. So depending on the protein, right, the folding, the secondary and the tertiary and possibly the quaternary structures, those don't happen on the ribosomes. Those happen in the endoplasmic reticulum and the Golgi apparatus. And so, right, the folding and if they're uh, attaching lipid molecules or carbohydrate molecules to make them lipoproteins or glycoproteins. And also one of the post-processing things is the removal of the start codon, because now you have the polypeptide chain, so you don't need that code anymore. And so the starting methionine is removed from the polypeptide chain, and that actually activates the protein. So, right, not only is the start codon, it's sort of the limiter. Once the methionine is removed, that activates the polypeptide chain, and so it becomes a functional protein, right? Whether it's a single polypeptide chain or made up of multiple. Does that make sense?
Yes, sir.
All right, so some of the details that I just gave you are not in the text, at least not in this chapter. And so I just gave them to you. Then there are three codes that code for that are stop codons, right? And so also, right, they are not coding for these guys don't code for amino acids. There are three of them, and those are ATT, ACT, and ATC. ATT, ATC, and ACT, right? And these guys are stop codons, and when the RNA polymerase, right, on the DNA, it reaches the stop codon, then obviously it stops, because that's the whole point. Okay, so the RNA polymerase makes the mRNA, reaching the stop codon, and we're done with that. And now you have your mRNA molecule. So the mRNA molecule leaves the nucleus via the nuclear pore. There's other processing that happens with the mRNA molecule, but since it's not mentioned over here, I won't go into it. Into the cytoplasm, right? And it goes to the ribosome, right? And so it attaches to the ribosome, and is read, right? And the proper term for that is translation. So it is translated, right? This process is called translation of the mRNA molecule, right, into the amino acid sequence, right? It is read as triplet codes, obviously. And so what happens in the ribosome is that the ribosome holds the mRNA in place, and then there's another type of RNA which is called tRNA. right, which stands for transfer. So the T stands for transfer. Transfer RNA molecules bind, right, and that's hydrogen bonding. So they bind to the mRNA. And so what's special about the tRNA molecule? What does it do? And bring in the corresponding amino acids. So the structure of the tRNA is unique. You can take a look at it in your textbook, but it has, right, the tRNA molecule, right, has a complementary. So there are several tRNA molecules, right? And so each tRNA molecule, let me add each over here, has a complementary triplet code, right? And that is called the anticodon. And what happens is that it matches with the codons on the mRNA, right? And it also has binding sites for the amino acid molecules. And so what happens is that the tRNA molecule, it looks something like this, right? And it's got this triplet code over here and a binding site for amino acids over here. And this is your mRNA molecule, right? And so these two guys pair up. And so, for example, this is the code for, let's say, methionine, right? So it's TAC, if I believe correctly, right? Or the other one that was tryptophan, let's say it's ACC, right? And so you've got ACC on the mRNA. And so what you have is UGG, right? I wish you can't make sense of that. So you've got UGG over here on the anticodon. And then you've got a tryptophan molecule, right? So tryptophan, that's not tri, it's just short for tryptophan, TRY. And so you've got this molecule over here. Now what happens is there are at least two tRNA molecules on, right, the mRNA at the same time. So you've got that and that and that and right something. And so, right, you've got another amino acid over here, and then you've got an enzyme coming in and it forms a peptide bond, right? So amino acids line up and, bond it together by whatever enzyme, right? They haven't mentioned it, I'm not going into it. And so that is the process of translation. The peptide bonds are formed. Right. Is that clear?
Yes.
So that's the second kind of RNA that you would find in cells, right, in your body. The third type of RNA is ribosomal RNA. Right, and so ribosomes are basically made of protein and RNA, right? And the RNA that is used in ribosomal RNA, right, is known as rRNA. So R standing for ribosomal. So that's it now, right? Protein synthesis, we already talked about, how that's working. And so again, transcription from the DNA to the messenger RNA. The messenger RNA goes, right, binds to the ribosome, the tRNA comes in, complementary codon, the anticodon, bringing in the amino acid, forming the peptide bonds, right? And so it is a continuous process. We've broken it down into steps to make it easier to understand, but right, it's a continuous process. And the last thing is talking about mass production of polypeptides. So the way we've taken a look at it is that a single mRNA chain is only read once, and it's got it's stuck to one ribosome, and like, no, that's not how it happens. So what's really happening is a single mRNA molecule can have multiple ribosomes attached to it at different points. Well, saying at different points isn't correct, because they're all starting from the beginning to, right, produce multiple copies of whatever polypeptide it is. And that's basically, so you take the entire process, and then you imagine, okay, fine, so that's happening with one ribosome. Imagine that there are 10 ribosomes, and so what you'd have is you have this use a straight line. So you've got this molecule, you got this, right, mRNA molecule over here, right? So this is an mRNA molecule, and what you have is, right, it's starting from here, it's stopping over here, so that's the end. And obviously this is the direction of translation. And so what you will notice is that you've got, so these are ribosomes, that's the large subunit, that's the small subunit. It doesn't exactly look like that, but anyway, right? And so what you would find is multiple ribosomes that obviously started at the beginning, and right, they're moving along, and you would find different lengths. So this guy will obviously have a shorter polypeptide chain, this guy will be longer, and then longer, and then longer still, and this guy will have the longest so far. And so there are different stages. And so what you're having in this illustration, for example, you've got 5 copies of that same polypeptide chain being translated at the same time, and so that's mass production. Does that make sense?
Yes.
And that's it. We've reached the end of the chapter, and we've completed right, section 2b. And yeah, so what's next? We've gene mutation. So yeah, tomorrow we'll be talking about, I think, gene mutation and inheritance. We'll see. I'll see what we'll do tomorrow, right? But for now, we've completed section 2b. All right. Any questions?
No, sir.
All right, then I will see you next class, inshallah.
Inshallah.